All right, hello everyone. My name is Michael Gomez, and I'm a postdoc at the IGI by UC Berkeley. And I'm going to be talking about the genome editing of cassava to eliminate toxic cyanogen production. This work was done in equal collaboration with Jessica Lyons at UC Berkeley and is funded by the Innovative Genomics Institute. So first off, what is cassava? Some of you might not be familiar with this. This is a staple crop grown by smallholder farmers throughout the tropics and subtropics around the world, primarily in sub-Saharan Africa, which produces over half of the world's cassava. You may also know this by the name of yucca, tapioca, or manioc. This woody shrub is actually very easy to grow. It has a number of advantages over other crops in that you can just take a stem cutting, take a piece of the stem, about 10, 15 centimeters, stick that in the ground, and 6 to 12 months later, you have a fully mature plant that you can harvest. In addition, this plant can grow for a long time without water. It can grow in nutrient-poor soils and can deal with the high temperatures of drought. Now, the real moneymaker here are the carbohydrate-rich roots. Farmers will harvest that, pull that from the ground, sell it at market, and it can be processed in a number of ways, from boiling to frying to drying in the sun. Unfortunately, this crop also produces a toxin, and consumption of insufficiently produced cassava, processed cassava, is associated with konzo, a paralytic disease. This is really a disease of undernourished and poor rural areas of Africa, which push people towards a monotonous diet and a high cassava consumption. Couple that with unsuccessful detoxification, so not boiling enough or not frying enough. This leads to a high cyanogen consumption, and in combination with the low sulfur amino acid intake, leads to upper motor neuron damage. In this condition, konzo manifests. This can manifest very abruptly, within a day, and it is incurable. There's no treatment for this. And the severity can range from mild, where a child can still walk, to moderate, where 1 or 2 sticks may be required to walk, to severe, where they cannot walk at all. And so what we'd like to do is tackle this, tackle konzo and remove these cyanogens. Fortunately for us, work has been done to identify the biosynthetic pathway that leads to the synthesis of these cyanogenic glucosides. 2 enzymes, 2 CYP79D enzymes, are involved in the conversion of valine and isoleucine into linamarin and lotostrilin, and the cyano groups are circled in red. In addition, cassava has been sequenced. We know its genome, so we have identified the genes that encode these enzymes. So where we'd like to go from here is to apply the CRISPR-Cas system to knock out these genes. We can target these genes using the CRISPR system, and this begins with taking the cassava plant and culturing a mass of cassava cells. We can then deliver the CRISPR system into it, where Cas9 will recognize an intact cyanogenesis gene, modify it, cut it, and during repair it'll be edited so that it'll be non-functional, and then no cyanogen production should occur. We can then regenerate a whole plant from a single cell, and hopefully we can distribute these plants and therefore eliminate the spread of konzo. Now, where we're at with this project is in phase 1. We've established tissue culture at the Innovative Genomics Institute, and we are moving on to bacterial-mediated delivery of the CRISPR system into these masses of cassava cells. You can see a mass of these cassava cells growing in panel A, and in panel B, we've delivered the CRISPR system into the cells along with a visual reporter, so we can see which cells have taken in the CRISPR system. At this point, we are regenerating these plants. And then we will move on to measuring their cyanogen levels to confirm that there is no cyanogen within it. And then on to phase 2, we'd like to apply DNA-free gold particle delivery. What this is we will coat gold particles with RNPs, with the Cas9 and the guide RNA, and shoot that like a gun into the cassava cells. This minimizes off-target effects because the proteins will eventually degrade. It's a transient process. In addition, this will not be regarded as a transgenic approach. And then moving forward from there, we would like to advance to farmer-preferred varieties. There's a wide variety of cassava varieties, and we would like to maintain that biodiversity such that farmers can have the flavor and the type of cassava that they have and also avoid any diseases that may affect a single variety. That's it. Thank you.